And great to talk after Marcel because that's a lab I have been looking up to their work for a long time. So actually, it's a great presentation. We don't actually need to talk more about what protein design is as well. So, I just want to say that there is actually a great similarity between man-made and nature-made systems in the sense that there is a hierarchy. We have heard about the hierarchy in a few talks, in keynote talks in this conference as well. But basically, they share the hierarchy of parts, devices, and systems. Just in our cases, the parts are biomolecules, whose functions and parameters are much more difficult to understand and engineer. So our central question is, how do we design bioparts, in this case, proteins, to effectively probe the inner workings of biosystems? That's sort of the reverse engineering. And at the same time, can you engineer utilities for biosystems, for instance, synthetic biology or therapeutics? So that's the central question. And what can we get from protein design? So usually you will start with a mechanistic hypothesis. You will have biophysical assumptions and models, and then you translate them into a mathematical model. And then you do some computation, and you got some designs. And hopefully these are optimal designs in the sense that they are the optimal based on your model. And then you try them experimentally. You found some gaps, some works, and some don't. And then you know exactly where to change your models and what hypotheses need to be changed. So actually, my argument for computational protein design is that it's actually a very important part of biology study, just like any other experiments. But it has to be very robust in the sense that, for instance, if you have a model and then the computation somehow can give you some good computational designs, and you know that it's not optimal to your model, but you don't know where exactly the optimal designs are, and then there are gaps between your designs and the optimal designs to your model, and then there are gaps between the designs and the experimental results, and then you don't know exactly where the problem is in the hypothesis. And then maybe you just didn't search long enough, maybe there are other reasons, so it's just hard to get that feedback between experiments and computations. And that's exactly the reason, like Marcel was saying, that we need exact algorithms that, based on the assumptions, hypotheses, and models you have, you should get the optimal designs, and then you can test them, and then you can examine the hypothesis. But there is a lack of efficient exact algorithms for multi-state protein design. Actually, the only one that's available is from the Donald group I'll briefly talk about. So what is our formulation of the multi-state protein design problem? You have a problem overall objective, and you are trying to find the optimal sequence S. Then this objective can be decomposed into a positive and a negative objective. For instance, you want the positive objective is a stable complex. The negative objective is to make sure that this protein fold. And in this case, you might want to make sure that this protein binds to the target, but not to the off targets. But then you may have sub-objectives. For instance, in this case, this positive objective is a minimum among many positive sub-objectives. For instance, you may have multiple backbone conformations, or you have multiple targets and off targets, right? So in this case, the relationship is an OR relationship, but you can also change it to max. For instance, the worst binder to the worst on target minus the best off target. So this can be done very easily in our case as well. But let's come back to this formulation and then ask, well, actually each substate problem is also a design and optimization problem. For instance, this is a fixed sequence, fixed substate, and different sidechains. So you have to do sidechain optimization. And so this is the optimization problem we are dealing with. Unfortunately, it's very challenging. And you can have different constraints. For instance, you can make sure that the stability is not too much worse compared to the wild type, and so on. So now let's come back to the challenges. Okay, how to solve this problem? Why is it challenging? So if we look at this one, though this can be regarded as protein folding because you have the fixed sequence, you are just optimizing the structure and the sequence. This is the single-state protein design, which is also very challenging, but not so much as multi-state protein design. So a long time ago, it has been shown that protein design is NP hard. That means you cannot have algorithms better than polynomial. That means your algorithms that can guarantee a global optimum is pretty much some exhaustive algorithms, but that need to be very efficient and smart. Otherwise, we just cannot enumerate all these combinatorials. So there's no exact algorithm exist for the multi-state protein design except COMETS, which was published in 2015. This is based on the dead-end elimination and the state A star, one of the first prototype exact algorithms for single-state protein design. So this is our framework called iCFN for multi-state protein design. So we start with an ensemble of structures for the positive state and ensemble of structures for the negative state, and then the mutable positions and the flexible residues around. Then you have some energy models, and then you have matrices. And then you just do a sequence-based and Rotamer conformer level tree search, which I will talk more about. And you start with getting a global optimum, and then you get also the best ensemble. You will get the top best sequences in their top best substates and in their top best conformational ensemble. And then maybe you want to have a higher resolution potential to rescore the top ensemble, and this can be incorporated, for instance, n-body, energy models, and something else, like our PBSA. So I will talk also about a couple applications. So let's talk about a little bit about the algorithm. So if you think about it, this is actually a hierarchical search. You start with a unnamed sequence XXX of 3 positions, and first you branch and you tell that, oh, whether the first position is availing or not, and then you keep doing the binary branching. At the end, you end up at a complete, fully defined sequence, like in this case, VAP. And then you end up having branching into different states and substates. And then for each state, you are doing a combinatorial optimization problem. In this case, the values are sidechain, so you are doing sidechain packing problem. So each of these substate optimization problem can be recast as a weighted constraint satisfaction problem and solved by the cost function network, which was actually pioneered by a French group in 2012. And then our contribution is that basically, if you represent this search space in a hierarchical tree, you can recast the overall problem as correlated weighted constraint satisfaction problem and modeled as interconnected many cost function networks, and then solved by depth-first branched bound using our novel bounds at various levels, sequence, state, substate, and lower. So I will now go to the applications. So the first application is a XRCC design. So we just want to make sure that it's a better binder that folds. So in this case, we'll compare to COMETS, but we have slightly different objective functions and different implementations. So we decided to look at the multi-state protein design with a single substate for either state. So it's a little verbose, but basically you don't have multiple substates for positive or negative. The positive state is a bound state, and the negative state is an unbound state. And then we are incrementally adding one and one until 5 design positions, and we'll keep the surrounding residues flexible. So that's the running time comparison. We're generating the top epsilon equal to 0.5 kcal per mole. So if we generate 1 or 2 mutations and keep the surrounding residues 3 or 6 angstrom to be flexible, you may have 9 or 16 residues. And then this is the size of the problem before the dead-end elimination, and this is after. And then this is the running time for COMETS, and this is the running time for reduced ISFN. That is, if we didn't introduce those narrow bonds and solved them as independent cost function networks on our trees. And then this is our fully interconnected cost function network. As you can see, it's a little bit 1 or 2 orders of magnitude faster. And then as you get in more and more positions, and then as you see that the memory problems, I think because we run these experiments in 2017, early 2017, so we didn't have the benefit of Osprey 3, it would be interesting also to see these cases as well. And our memory use is at most 80 megabytes in these cases. And as you go to higher epsilon cutoff, you're having more ensembles and you have more advantages. So now let's, so there are some theoretical reasons that why our depth first branching bound is more powerful than the A star used in comets, and I'll skip that very briefly. So now let's talk about second design. So this is a T-cell receptor specificity design for immune therapy. So this is actually one of the first T-cell receptors developed for melanoma. Here we are trying to make it a specific binder to a MHC peptide complex. And this peptide, the positive state is a peptide which is marked AAG peptide, and then negative state is the marked ELA epitope. And the substates are actually global backbone ensembles generated by MD simulations. And then the substate functions are folding of the complex at the beginning, and then the binding affinity at the end. Initially, it was just the Coulombic electrostatics. At the end, it was MMP-BSA. And the design positions are based on a previous study, 4 positions. We did up to the triple designs. So here is the design efficiency. Because we couldn't run comets in this case, even because these cases are even larger. So we compare the reduced iCFN and iCFN. So as you can see that this is the post-dead-end elimination size. So these are up to 10 to 56 in this case. But reduced iCFN only search states for like 10 to 4 and 10 to 3. And then, of course, they have to search all the sequences because there's no interconnection. When you introduce interconnection, you see that the space, the effectively searched space, is even 1 order of magnitude smaller. So it's still just like 10 to the 3 in the space of 10 to 56. And we don't have to search many sequences, actually, if you compare to the reduced iCFN. We only search 3 to 5 sequences, and the running time is only seconds. And then this is just for the global optimum, right? And as you do the double designs and the triple designs, the number of sequences you have to search is actually very, very small compared to the number of sequences in the complete design. So as a comparison, you will want to ask, well, what is the speed up due to the interconnection as it related to the problem complexity? So as the problem complexity increases, this is in the log space, and then the speed up also increases. So this is the speed up in the sequence space, and this is speed up in the running time. So there's more benefits from the interconnection when the problem complexity increases, actually. So what are the accuracy? So we compare to the experimental results. There are 6 known mutants. And there's this delta delta G, which is a relative binding affinity to the wild type. So the energy calculations, the correlation, our correlation number is somewhere between Rosetta and the Rosetta mean, meaning the Rosetta mean is considered a local minimization. And a slightly backbone minimization. And then delta delta G is the difference of delta delta G between positive and negative states, and then you use whether it's negative less than 0 or not to predict whether these designs are AAG specific. So using Rosetta, you will get 4 of the 6, and then but you incorrectly classify the other 2 as false negatives. And use iCFN, you will get all 6. Actually, there are 7 correct designs, but there's one that's not got by any of them. That's G28N, and there's no false negatives. And then what about the characterization of design space? Because we are providing the guaranteed top sequences in their top states and top ensembles. So they actually tell you the energy landscape and individual positions designability. And we found that position 98 on the alpha chain having much less sequence solutions than the worst binding affinities is much less designable, and that's agreeing with the previous results. And then if you talk about what if I only use the fixed backbone, just the crystal structure, as you can see that you will only get 3 of the 7 instead of 6 of the 7. Actually, these backbone conformations are utilized by different positive or negative states and by different sequences. So backbone conformers will provide much more diverse solutions and higher coverage in protein design. And then also it reveals the mechanism. So we correctly predicted that introducing bulkier hydrophobic residues in these positions would improve binding specificity. But why? So we look at our models by iCFN. So these are the proteins that we designed in blue. And then this is the residue that's mutated into isoleucine. And the cyan is a peptide, and the wheat is MHC. So the substitution is from glycine to isoleucine, so it's much bulkier. And this is AAG, and this is the ELA. So the N-terminus of AAG is alanine, and the N-terminus of ELA is glutamic acid. So upon mutation, the previous hypothesis, and it's validated in our case, is that the bulkier isoleucine 28 forms worse van der Waals packing with the ELA peptide, but not so much with the AAG. But something else we found was that actually it also suffers from the continuum electrostatics, because as you can see that once these bulkier hydrophobic residues in place, it actually blocks the solvent accessibility of this glutamic acid. We're actually standing in the solvent and looking into the interface. But here is an alanine, so it doesn't pay more desolvation penalty upon the mutation. So at the end, I'd like to summarize that we provide an exact algorithms can help you understand the mechanistic hypothesis and feedback between experiments and models. And this is the first exact algorithm for multistate protein design that is not based on dead-end elimination, A*, and it can solve large-scale design problems of unprecedented sizes to previous exact methods. And we have seen much boosted efficiency and competitive accuracy compared to the state-of-the-art methods. And the consideration of backbone global flexibility leads to more diverse solutions in protein design. So this is the reference. It's just accepted to ECCB, and it will appear in a month or so. And we have currently, I only have one student working on this one, so it's now provided as a binary, but we plan to make it open source very soon. But if you are interested in solving your problem, even if it's not a protein design problem, but that's a valued constraint satisfaction problem, you can just generate your results in a table and then run this problem. So it can be even not a protein design. It could be just a weighted constraint satisfaction. So at the end, I'd like to thank my student who has been Mustafa, who has been the boosting force and the big reason that we have made this possible. And also I'd like to thank the Bruce Donald group for the Osprey and the years of inspirations on developing exact algorithms. We also had help from Dr. Mark Helland on how to use Osprey, especially comets. Actually, I'd like to say that comets is also a very important update on the Osprey 3. And I'd like to thank our founders from NSF and NIH and A&M. We have positions for PhD and postdoc, and we're very much looking forward to your joining. And this is our website. Thank you. Yeah, that's a great question. I did tell Mustafa like more than a year ago, but unfortunately I only have one Mustafa, and he's a little hesitant into CUDA programming. But I already had the acronym for my next algorithm. That's Pi CFN. Pi stands for parallel. Yeah, but great question. Hi, Marcel.
Thank you so much. I thought that the talk was absolutely fantastic, and it's very exciting to see new provable algorithms coming around.
Thank you.
One of the questions that we had is, in the realm of continuous flexibility, do you think introducing that would improve your results, and are there plans to go on that route?
Yeah, that's a great question. Actually, here we didn't consider the continuous flexibility of the rotamers, and we didn't consider the flexibility of the backbones locally. We thought the global would be better coverage, but another reason is that it's already very successful in your lab, so it's very easy to just implement the voxel-based local flexibility to the sidechain or global or the local backbone conformations, because it's just one additional level or min and max, that's right. So yeah, it's conceptually it's not difficult. Thank you.
Thank you.
Great question.
Let's thank Jan for the nice presentation.